Ahora. So, okay, good morning for everybody. And now we have our third webinar from our RSG Colombia. And today we have a pleasure to present Gloria Giraldo-Calderón. She is a biologist from Universidad del Valle in Cali, Colombia, and she holds a PhD from Purdue University in West Lafayette, Indiana, and in USA. And currently, Gloria works in VectorBase, that is a highly recognized database that provides genomic, phenotypic, and population data to the scientific community to invertebrate vectors of human pathogens. And now in VectorBase, Gloria works as a scientific liaison, and she's the outreach manager. So welcome, Gloria, and please go ahead with your talk, Arthropod Genomics, data tools, and online resources. Thank you, Gloria.
Thank you, Jacid, very much for the invitation, and thank you, everybody, for being here today. I want to make this very participative, so please, I don't know, because I have the screen of the presenter, so I'm sure for you it's different. Locate where is the chat, because I want questions. I want you to interact. So we are going to give it a test. So everybody just say something, just say hello or anything. So I know that everybody has find how and where to ask questions. Okay, thank you. Excellent. Excellent, guys. Very good. Okay. What are we going to talk about today? Arthropod genomes and transcriptomes available. I'm going to talk also is the omics data that you have when you're interested in arthropods. I'm going to talk about open access databases and tools specific for arthropods and others that are more general but also include arthropods, but only going to talk about free resources, and also about data access and publication policies. Oops, that's not looking good. Where to find arthropods, arthropod data and resources? Well, arthropods are the largest and most diverse eukaryotic phylum with more than 1 million species. So if you're interested in them, where can you find the omic data? There are the most important archival repositories: EMBL-EBI, GenBank at NCBI, and the Database of Japan. These three databases, as you know, communicate daily. And when you have an accession number from any of these three databases, they work in any. In addition to that, we have specific databases. And here I was going to show you, well, Jacid, if I share my screen, were you able to see the presentation directly from my computer, you think? Let me try that. Start screen.
Yeah, I think that is possible if you share direct your screen, and maybe, yeah.
Okay, is that better?
Yeah, it's better.
Okay, good. Okay, so there are specific databases, and here are only 10 examples, but there are many more. We have AphidBase, Apollo, which is a tool in which you can, you have the genomes and experimental data, and with that, you can manually annotate a gene. What that means? You sequence a genome, and there's an automatic way to generate all the genes, of course, using RNA-seq experimental evidence. But that with the technology we have today, that doesn't replace a human actually correcting the genes. If you are the expert, for example, in the GPCR proteins or olfactory or gustatory proteins, you come to Apollo and you manually check that each one has the start codon, the stop codon, that the splice sites are correct, all the exons and introns and everything. And Apollo is an online tool, which means that whatever you are doing is everybody in the world can see it in real time. So we are not wasting time about annotating genes twice. And this information later gets updated to the official gene set. BUSCO, I'm sure many of you know about this tool. This allows you to find common orthologs between all the arthropod genomes. So you know, is my genome complete? Was my genome sequence, my assembly and my gene set have at least a core of genes that I should have? Hymenoptera database is more like, can you see my cursor, right? Can you see my mouse, Jesse?
Yes.
Okay.
Yes, it's okay.
Okay, so AphidBase number 1, number 4, there's Hymenoptera database and VectorBase, for example, are more species specific and oriented. For example, VectorBase is more for species of medical importance. Hymenoptera, the names gives away, Hymenopteran species. This one here, I5K of the workspace up now, is for genomes that do not have a home. And you do not have the resources to construct your own website, but you want it available for everybody and use all the tools that they have. This is a great resource. You contact them and tell them, my genome does not has a home. I want it here. But remember, your data has to be first in the archival databases, and later any other of the other databases will take it. That's the main requirement. Must be here first. Lepbase, Panoptes, this one is a tool specifically designed for the Anopheles gambiae genome sequencing project. They started with 1,000 Anopheles. Now they are going to more than 10,000, but I'm going to talk about that later. OrthoDB is to find ortholog genes, groups of orthologs for your species of interest. We are going to take a quick look at that at the end with the demo. Ensemble Metazoa. Ensemble is a family of websites, and there is one that is Ensemble for humans, but there is also Ensemble for plants, for virus, for bacteria, and Metazoa is the one that includes arthropods, and I'm also going to give you a quick demo at the end. And VectorBase, as I said, for species of medical importance for humans, I'm also going to give a demo at the end of how it works. Why this asterisk? Because where can you find all the other tools available? If you don't know yet, I strongly encourage you to look at this journal, Nucleic Acid Research, the NAR journal. They have 2 special issues per year. One published in January is the database issue, and one published in July, the web server issue. And here you'll find databases and tools, tons of them. And these publications have been going on for many years, as you can see there. Okay, so the data. Where are the genomes sequenced? The first one, of course, the fruit fly, Drosophila melanogaster, year 2000. But today, there are more than 360 species, and these represent 36 orders and about 150 families of the 3,000. So this is not much. There is a lot of species bias here. So these species are mostly from hexapods, mostly from holometabolous insects, and especially from 2 orders, Hymenoptera and Diptera. Where can you find this information? Of course, you can just go to NCBI, as I just said. But there is a nice table and overview here if you follow this link that will show you all the different species. And it will give you the order, the family, the species, the common name, and where is the database where that species is living. This effort is I5K. The idea is to sequence 5,000 arthropod genomes. So far, we have 360. How do you become part of the I5K? We are all part of it. If you already sequence a genome, if you're using the data, you are I5K. And they do an annual meeting. I strongly encourage you to attend AGS, the Arthropod Genomics Symposium. They meet there. The meeting is in 3 different universities in the US, the University of Notre Dame, the University of Illinois and the University of Kansas. And there you will learn and share what's new every year. Transcriptome sequence, there is a big project called One Kite, and they actually intend to sequence 5,000, sorry, this is a typo, 1,000 insect transcriptomes. So this is arthropods and this is insects, but not 5,000, 1,000. They started in 2012. And if you want to know the list of species that already have been sequenced, you can follow this link. And unlike this one, where there is a bias for these two orders, here all orders of insects are represented. So this is a great resource. I'm going to give you a look of the first 10 years of insect projects. At the beginning, most of the things that got sequenced was important for genetic status, development, or molecular biology things, or a species of medical or agricultural entomology. At the beginning as well, it was, you knew that having a genome will give you the front page of a journal. So for example, here's a science journal, when they published the Drosophila genome, and here the malaria mosquito, Anopheles gambiae, in Science as well. These are the species and the years. Drosophila melanogaster, the fruit fly, in 2000. Bombyx mori, it actually got 2 genomes in 2004. Another Drosophila in 2005. Honeybee 2006. Here I have an asterisk with this mosquito. This is the vector of yellow fever, dengue, Zika, and chikungunya. The reason why I have this is because the first genome got sequenced in 2007, and the next one, which is now in chromosome form, actually in 2017. And this is something that is not the only species, but I'm just showing you. For many species now with better technologies, we now have resequenced them and are going from supercontics and scaffolds to actually having chromosome assemblies. Drosophila, later, in addition to these 2 species, later got 2 more species in 2007, trivolume, 3 species of Nasonia, and the list goes on. From here, so this is the first 10 years. From 2010 to 2019, it's been an explosion of insect genomes. But look how small steps we took every year to get to where we are today. Genome groups. For some genus, we have the luck of having more than 1 species. For Aedes, we have 2. For Anopheles, 19 species. Apis, 4. For this is honeybee. For ants, we have for this genus we have 2. Drosophila, today there's 30 species, and I'm sure by the time we finish this talk, probably will be 1 more. Glossina, this is a vector of a disease in Africa. 7 species. Butterfly, this genus, 16 species. But also people, in addition to sequence species from the same genus, they also sequence related species. Why? For example, when they sequence this fly, Glossina, they sequence 7 species. In addition, they sequence the housefly and the stable fly. And this is useful for comparative purposes. These are some flies, the vector of leishmaniasis, and they sequence 1 species from the New World and another from the Old World, 21 species of aphids, 19 of ants. And there's also big flag projects. For example, 30 strains of this mite, Tetranychus, 1,000 genomes of Anopheles. I'm going to give more details about that. 200 hemipteroids, 5,000 Daphnia genome projects, and many more. This is just a list of the things available out there. This is just an example. So what people is doing when they sequence more than 1 genome. MalariaGen, the Genomic Epidemiology Network, started a project in 2014. The idea was to sequence 1,000 Anopheles gambiae. Now, I'm sure you all seen this in bioinformatic presentations, and I think the cost of sequencing have decreased so much that with the resources that they started, now they can do more. And actually are planning to do 10,000 individual genomes per year, and they are going to include other Anopheles species. These are complete genomes. Why they want to do this? Because now they want to do analysis of variation and polymorphisms. They want to look at natural populations, and they are collecting in 13 different countries in Africa. Now that they expanded the project, they are looking at countries in Asia. They want to look at population structure in time and space. With the new the new project are actually looking at museum specimens. So before, I'm sure you're aware of this, insects are small. And when you sequence an insect, before you had to take a pool and take, I don't know, 5, 10, depending of how small they are. When you were going to do the assembly, that became very complicated because if you are working, for example, with a diploid species, so you have the chromosome from the mom, the chromosome from the dad, and when you're going to do an assembly, the haplotypes will make it very difficult. Now, this project, they are being able to use, now with the current technology, you can use single species, even for a mosquito, which is really small. But now for this second part, which is using museum specimens, I'm sure nobody in a museum will like if you destroy a whole insect. Just with teeny little parts of these insects, they are able to sequence them. And the reason why is because they want to look at the spread of insecticide use before insecticide was widespread. And the fact that they are able to do this means that for your species of interest, you can use a single specimen. And we can discuss this in the Q&A part. What are the advantages and disadvantages of use a single individual? Well, we'll come to that later if that's your issue and your species is small. They are also looking at the relationship of the variation with biology, ecology, and epidemiology of malaria. Having one genome is great. There's many things and questions you can ask, and having more than one from a single species allow you to ask a whole different set of questions. In this case, variation in populations. They are also looking at evolution, gene duplication, and selection patterns. I also told you that there is transcriptomes available, and this is a paper that came in 2014. So for this one kite project, in this initial paper, so I told you, they already sequenced more than 1,000 species. But for this paper, they only selected 103 from, they included hexapod species, all the orders, and they are looking at, they also included, this was not sequenced by them, but they included 14 species with their genomes and 27 transcriptomes that were already available. And this is one of the figures from that paper. And in these three, they have constructed these using more than 1,400 protein coding genes. This data is there and is available for you. And we're going to discuss at the end. How can you use this data generated with projects like this? Maybe you're saying, well, there is definitely more than 1,000 transcriptomes. Yes, there are. I was just telling about the full transcriptomes. There is other type of transcriptomes, the ones that we sequence to look for differential expression. Which conditions can we test? You can test for males and females, different life cycle stages of the egg, the larva, the pupae, the adult, meal source. I want to test what happened with my insects when they eat blood, when they eat sugar meal. Body parts. You can dissect your species. So for example, you are working with a spider that's being parasited by hymenoptera, and you want to look what happens in the brain of my spider when it's behaving like a zombie because of this wasp. Or take the legs and the antenna of an insect. You can test different treatments. For example, you apply insecticides to some insects and to some others not. So, and from these differential expression transcriptomes, there is definitely more and more data. In addition to arthropod genomes and transcriptomes, we also have proteomes. Again, this could be for specific tissues and organs, and you can test for different experimental conditions. Also have control and treatments. However, there is definitely less data for this. Doing a proteome is more expensive, and there is still a lot of standardization to be done. But that data is available. Okay, so I was telling you. So you're in a lab in which is not easy or accessible for you to sequence a genome or a transcriptome or a proteome. That's definitely something that is not in your reach. For example, today, to sequence a single mosquito using PacBio, for example, could be around $20,000, and some people do not have that. Probably is less with other technologies, but let's give that as an example. So can you use that data? I'm going to talk about, you find that data in a database, and you have your specific question or hypothesis. There's two scenarios. If the data has been published, if there is a genome or transcriptome paper out there, it's fair game. You can publish. Let's say, for example, you're working with an arthropod genome of about 20,000 genes. I'm sure in the genome paper or transcriptome paper, they have not looked at the 20,000 genes, but it's already published. So it's free to use, and you can do whatever you want with that genome. However, if you find a genome or transcriptome or proteome in a database, but there is not a paper associated with it, you cannot use it unless you ask permission to the consortium leaders. For example, in the OneKite website, they provide a link and a contact form, and they said, if you want to use our data and it's not being published, this is how much you can, and then they give you guidelines. If you want to do things with a gene family across all 100 species and it's not being published, you cannot do that. But if you're going to look at 3 genes in 4 species, there's something definitely that you can do. So bottom line, genome published or transcriptome published, you can do whatever you want. If it's not published, contact the authors of the data, ask permission, tell them what do you intend to do, and they can tell you yes or no. And if it's not being published, they may ask you to include them as author in their paper. But if the data has been published, you just need to cite the paper. You don't need to include them as authors in your paper. And all these guidelines of ethical behavior of use of omics data is part of the Fort Lauderdale Agreement from 2003. What type of analysis can I do with the data? Anything, your creativity is the limit. You can do taxonomy, systematics, phylogenetics, evolution studies. You can look at gene flow, presence or absence of genes. You, for example, are looking for the opsin genes in mosquitoes, and you take Aedes aegypti, Anopheles gambiae, and Culex fasciatus, and you're looking at the ultraviolet, short wavelength, and long wavelength genes, and how they are present or absent in your mosquitoes of interest, depending on the behavior of the mosquitoes. Am I diurnal and diurnal? You create your project and your question with whatever data is available. You can look at presence or expression of genes that could inform natural history, insecticide resistance. You can look, for example, symbiote or vector relationships with virus, bacteria, or parasites. You can look at population genomics, looking at biodiversity, extinction, species expansion or invasion, population history. So do not think that when you're taking this data, you can only look at molecular aspects per se. You can look many things. And here are some examples. For example, if you're looking to support control strategies, you can look, for example, for your species of interest, where is if there is, for example, more than one individual sequence, you can look at frequency alleles and susceptibility or resistance to insecticides. You can look for insecticide surveillance. If you're looking, for example, virus-vector interactions, you can look at the immune system and how it responds to infections. So it's being infected with different strains or different species of any parasite or pathogen, or the impact of Wolbachia and a virus infection in an insect. Why these genomes and transcriptomes and proteomes need a database. Why do we need the tools? Because if you just have the genome, for example, sequenced, and you have an assembly, this is all you're getting. And with that, you cannot go very far. You need a gene set prediction and manually work with that. And you need a home where that genome will live, and you need tools to analyze it. So if you think of the databases as the home where the data will reside, each one of the rooms of the home will actually be different tools and resources of that database to make sense of this data that otherwise you will not be able to work with. And now I'm going to do a quick demo, but before I do that, do we have any questions so far? Jesse, any questions?
Hi, Gloria. I think that it's time for questions. Any questions, people?
Before we start with the demo?
Yeah, please go ahead with the demo, and maybe at the end of the webinar, we are going to ask again about questions.
Okay. I'm going to do a demo of 3 databases, VectorBase, Ensemble Metazoa, and OrthoDB. Probably you're saying, oh, that's not the one that I want. From the list, I want another one. Once you learn how to use one database, you pretty much it will be easier for you to get to use another one. So VectorBase, for example, it has here a search box. This is VectorBase Google, and you can type many different type of keywords, a gene name, or gene ID, or interpro. But I strongly encourage you to use a gene ID. Why? Because, for example, you type a word of a gene and you don't find it. It doesn't mean that it's not in the database. It just may mean that somebody has not named the gene in the species you are looking for. For example, you look for opsin genes in body loss and you find none. No, the genome can have that gene, but nobody. If you publish a paper, you need to submit the name to a database, because the databases are not going to grab the data from your paper. You need to actively go to the database and tell them about your data. We have the data divided in genomes, transcriptomes, and proteomes. And I'm not going to talk about these, but we also have population data. People that is doing studies in the field and looking at variation, insecticide resistance phenotypes and genotypes, population abundance, and things like that. We have a lot of events. We also have webinars, we give workshops, and attend meetings where we can meet with our users. These are the tools that we have. As I was saying, this is the home where the data lives. And we right now have 20 different tools that people can use to interact with the data. Apollo, as I was mentioning to you, they want to annotate the genes. So, for example, if I go to Aedes aegypti and I click on strain, you can notice this is the most recent of the gene sets, was updated in December. If you're just starting with a database and you really don't know what to do or where to go, for example, with VectorBase, I suggest you start with this. Find a data display. The backbone of VectorBase is the genome browser. And the genome browser has 3 different types of visualizations. It has genes, genomic location, and variants. This is mostly SNPs. And you select your species. We have 40 genomes. I live here in Anopheles, Gambiae. And this is VectorBase gene ID, what I was telling you that you should use to query the database. Because this is Anopheles, Gambiae, so it has these letters here, A, G, A, P, and a 6-digit number. And if you don't know the VectorBase gene ID, just run a blast inside VectorBase with the sequence and you will obtain it. Look what it's telling me. VectorBase has 47 different views for that gene and is divided in sequence and structure, expression and regulation, transcripts of proteins, comparative genomics, and variants SNPs. And if I scroll through all of this, it will give me details and all the tables and figures, and all this you can use in your publications, in your poster, in your thesis. Just take a screenshot and use them. Genomic location. So I'm telling, I want to go to chromosome 2L and in this base pair range and see what VectorBase has to offer me. Here we found 13 views. And you can, again, explore all this information and see how can you use it. And the last one is the variants. This is something that I do not encourage you to use in your papers because this is a temporal ID. And this is telling you that this SNP, could be an SNP, we'll see, is located in chromosome 2L, and this is the base pair position and the reference and alternative allele. And we found 29 different views in VectorBase. And notice how this here change as well, population and individuals, and all these different ones. Let's choose one. So it's showing population and individuals. And look, for example, these spikes are showing here from all these different population, where is the frequency of alleles. But you want to actually go to the VectorBase page where you can find this. So yes, this is a SNP, it's a missense. It has a phenotype attached, a publication, whose sequence I was telling you about, the Anopheles 1000 Genomes Consortium. If you see the dotted line, it will give you more information. This is Anopheles colucii from Angola, and this is the frequency of the alleles. And you can download all the information on CSV format and work in Excel or any other program. So that's to get you started with VectorBase. I encourage you to use that tool. And it has, the usual BLAST and Clustal and any other tool. Any questions about VectorBase? If somebody has any question, anybody that works with medical entomology or vector biology?
People, any questions about VectorBase? Okay, if no questions, please go ahead with your second database.
The next one is Ensemble Metazoa, and here, let me see. So as you can see here, I was telling you at the beginning, this is a family of databases. So you have Ensemble genomes, bacteria, protists, fungi, plants, metazoa, and vertebrates. And because we're interested in arthropods, so I came to Metazoa. This is the full list of species available. And you can organize them and sort them based on name, classification. So if I click on classification, I have annelids and chelicerates, and look, here are the insects, beetles and flies and mosquitoes and Drosophila. What are the tools that you have available? Hammer, you also have in VectorBase, BLAST, BioMart, all these tools also there, and the Genome browser as well. And here, they are very good with documentation, and they have an excellent support group, and the same with VectorBase. If you have any question, always look for the contact us. Remember that always behind this website, we are real human beings ready to help you, and we are a service provider. So anytime you email, in somebody in these databases, I'm sure you will get an answer between 24 and 48 hours. What is BioMart? This is probably one of my favorite tools. If you are not very familiar or comfortable extracting data from a genome, and you don't know how to code, and you don't know, and you really want to do genome-wide analysis, this tool allows you to ask questions just with clicks, no scripts or anything. You need to choose the database. So there are two, genes and variations. Remember, variation in this language means SNPs and related. We're going to select genes and later a data set, which species you want to do. For example, want to do this mosquito right here. And Biomark allows you to do, to construct your query looking at filters and attributes. Let's go to filters first. For example, you can select a gene and provide a list here, but there is many different things that you can ask, but let's pretend you can either type it here and just a list, or upload the file, just a text file, txt will work fine. And you can say or because I have not provided anything, so we'll just ask genome-wide in this case. But you can, filter your query, or you can say, I only want to know things about, protein coding genes, for example. Or I only want to know about genes in chromosome 2L. Or I only want in this superscaffold. Or I only want to query things about specific genes with a specific Go term. You choose. In this case, I'm just going to leave it like that, so it will be genome-wide. Attributes. What do you want to know about those genes? Well, I want to know what is their name. I want to know what is their function. Where is that? Protein. Description is their function. Let's give count. Well, this is going to take a while because I didn't select for some species. So it's looking genome-wide. And of course, because I didn't filter, so it's giving me 16,000 hits over all the 16,000 possible ones. Show me the results. And by default, it's only going to show us 10 rows of data, but you can download what you obtain in CSV format or XLS, and you can work that in your computer. And these are links. If you click any of these, it will take you to the Genome browser. So for example, I open this one. It'll give you the gene and the transcript. What is the difference between the gene and the transcript? Each gene can have more than one transcript and more than one protein. Because remember, if a gene has, for example, 3 exons, one transcript can only express the first and the third, and another transcript can only express one of the exons. So that's how Ensemble differentiates between them, and VectorBase as well. So you will find an R if it's a transcript and a P if it's a protein. And we use the letters of the alphabet to name the transcripts. In this case, these are A because it's just A single, or transcript number 1 is giving you the name and the function. Remember what I told you, when you're looking at the databases, you may be putting a gene name that you found in Drosophila, and you come to the database and you use that as keyword and you didn't find your gene. It doesn't mean that doesn't exist. Look, it's just that many genes names are missing. So that's why it's always to use the gene ID as the query instead of the gene name. I click on one of those and I come here to the Genome browser. What do you find here? A gene tab, transcript tab, and location tab. Every time I'm in one of these tabs, these menus here are different. And remember, if you want to change species, these are the favorite ones, and all the other species are right here. Every time you click for here in summary, but I can go to, let's go to ontologies. I want to know what information is there for this gene. So it's a protein, and remember, cellular component, molecular function, biological process, and I will give you the links to external databases like UniProt and Ensembl. And you can see here also the links to RevSeq. And you can change here, for example, right now we are in gene, but you want to look at the transcript. And notice how these menus here change. And now I want to see protein. And protein summary appears here. Location. The main thing that this is showing you, if the species you are working with has a chromosome assembly, it will be here at the top. But this species, for example, well, this is the older Aedes aegypti genome. This is L3. Right now we have L5. L5 is in chromosome form. L3 is in supercontic form. So here's showing that. And notice this red window here. This red window is what we have expanded here. And this other red window here is showing here even more in zoom. And we are looking at this gene. And look, these are the UTR region. And in red, in solid red, you can find the exons. These are the introns. And again, this is the UTR. And you can move around and you can say, well, this green gene here is the one you're seeing at the bottom. I want to look at this other gene. And you can jump to that region, for example. Okay, so we look at two tools already. We look at here in Ensemble Metazoa, we look at Biomark, and we look the genome browser. Yasid, is somebody has any question about Ensemble Metazoa?
I think that no questions.
Okay.
For now.
There's many other tools, and I said you can go and ask their help desk. The final one that I want to show you is OrthoDB. This is different from VectorBase and Ensemble, in which it's not the home of the genomes, but it's a resource to use when you're looking for orthologs of your genes and to construct some analysis. So you can, for example, type a gene and look how autocomplete is providing me with suggestions. Which keywords to use? Look at here, this question mark is giving you all the different identifiers and keywords and annotations and phenotypes that you could use. For example, you can use interpro domains. You can use words from IDs from other databases. Look, for example, UniProt, Ensemble, Antres, KEGG, Unigene, GenBank, RevSeq, Interpro, Genontology, and even the databases, AphidBase, BeetleBase, FlyBase, Hymenoptera, Lepbase, VectorBase. So this is a great resource. And when you email them, they are actually pretty good at coming back with an answer of how to use the website if you don't understand something. And for arthropods, I think they already have more than 100 arthropods. So this is a really good resource. You submit your query. Oh, sorry. So before I did this, the reason why I only got 24 groups in Hexapoda is because I filtered this and it's remembering my previous query. So here we have 170 arthropod genomes. And of these, 152 are hexapods, and is remembering that I have selected that. But you can select whatever you want. So that's why my results came like that, with 24 groups from Hexapod. Here in this side is showing you this orthologous group, and is for opsin, for C-opsin, for RH7-like opsin, and different types of opsin. RH3, blue sensitive, ultraviolet. In the other side is telling you how many genes and how many species are in that group. You can download this data, all the sequences in FASTA, or you can download the data as tab delimited. So you can work with this in Excel, for example. Let's wait until that loads. Here, this one is finished. This is the data that you can load and tab delimited, and these are all the FASTA sequences. What is this telling me? There's functional descriptions, there is evolutionary description, and here are the orthologs. So it's telling me for the query that I constructed, which species can I start to look at that have that gene that interests me. The good thing about them is that, look, all around the website, you're going to find, how to construct the query or what the different things that they have in the website mean. And, sibling results, how to export the data, how to interpret the data, upload, retrieve, bookmarks. So I strongly encourage you to use their website for your analysis. For example, I was showing you at the beginning that there are some species that have more than one species in their genus, or even not in the same genus. Like, for example, the case that I showed you of the 7 species of Glossina that they also sequence, the housefly and the stable fly. But sometimes you don't have those resources available to do comparative genomics. So OrthoDB is a great database in which you can actually come and look for some homology analysis and get great data for your research. I think that's all I have for today, Jacid. So I'm now open for questions if there are any.
Hi, can you hear me?
No question? Jacid, I cannot hear you if you're talking. Jacid, again, if you are talking, I cannot hear you.
And now? Hi.
Yes, I can hear you now.
Okay.
Any questions?
In the video, so, okay, we have a question from Carlos Alberto Cermeño, and he said, which is the best database to make comparisons in genes of insecticide resistance in Aedes?
So comparative genomics in Aedes aegypti?
I think so, that is, yeah, Aedes aegypti. Insecticide resistance related genes in Aedes aegypti.
Well, there is 2 websites, so both VectorBase and Ensemble. Well, actually, these 3 databases are actually the ones you should look for. So there is a part in the website. Where is it? It's called, I think I closed it. Well, if you go to Ensemble website, their pipeline is called Compara, and they actually have published how they have compared all the genomes, and that's a great resource. VectorBase, if you're, well, I said I was not going to talk about this, but your question is related with that. The population resource that I said has data about that.
Sorry, Gloria, can you please share the link of Compara?
Oh, sure.
It's just sharing in the chat.
You're still looking at my screen, right?
Yeah, right.
Okay. Here for Aedes aegypti. Or here. Aedes aegypti. This is the front page of the Genome browser, and look, they have a dedicated section for comparative genomics, and I strongly encourage you to use this. And in VectorBase, you can come to population biology, and we have a map. And you're looking for genotypes of insecticide resistance. And here, this map can take alleles and locus from anything, but it has started just with exactly with insecticide resistance. So here you can play with the website, and if you email me at info@vectorbase.org, I will be happy to give you more details about your query. But this is the tool that you want to use if you come to VectorBase. Any other question, Judith?
How is your email for doubts? Is
my email, if you have any
can you
questions,
please write there in the chat?
Yeah. This one. If you email to info@vectorbase.org, that's me. So for those of you that speak Spanish, you can email me in Spanish, that's fine, or in English here, info@vectorbase.org.
Okay, I am also sharing here in the chat. So we have another question from Karen Loaiza. Can you explain a little bit more about how to get genes not previously published? Like, can you search for a random gene now?
I'm not sure if I understand. Why the distinction? So you want to know if you're looking for a gene and you want to know if the genome has been published or not, or are you talking about when genes do not have a name assigned to them yet?
Yeah, good point.
Which species are we talking about, or which database, or can you provide more details, please?
Because she's asking about a random gene now. Can you search for a random gene now? But you are right. I don't know if a random gene refers to a published or unpublished, or what does she mean with random?
Which species, or, well, okay. If it's random, so I'm going to give a random answer.
Okay, Karen said for the ones without a name.
Okay, good, excellent. So it's as I show you, both Ensemble and VectorBase have BLAST. So when you do not have the name for that, the best thing to do is, just paste here the sequence of your gene, and in the results, it will give you the gene ID. And with that, you can start. So if you don't have the gene name or the gene ID, start with the sequence query, and that will give you the results of the gene ID in the gene ID format that you need.
It's okay, Karen? Okay.
Is that okay, or are you looking for something else?
No, she said that it's okay.
Okay.
Okay. Any questions? But maybe to finish, I have a couple of questions for you, Gloria.
Sure.
According to your experience working on VectorBase, what do you think that is the main challenge for the creation and management of this kind of big databases?
Mm-hmm. Well, the big challenge is this is a free resource, but free to the users, but it's not free to maintain. As I said, behind this website, there is humans. In our case, there are 25 of us. And I know some databases are now charging. I think in plants area, Arabidopsis, if I'm not mistaken. But I don't know if in our area you could do that and charge a fee. And yeah, you could differentiate that for low income, middle, and high income countries. But I will say that's the most difficult thing, creating the database, maintaining the database, all that requires human and economical resources, because you need the servers to store the data, to maintain the website. You need to pay the salary of the people doing
Sure.
To do, to provide this is a service, you're providing a service. So you need to have a help desk. I would say the most difficult thing is just to get a funding agency that will support this. So to the end user, this is a free resource. And that's why if any of you have tried to create your own website, at some point, you know, you run out of time or funding and you have to leave it there. So that's why I think it's a better idea to have a central website with all the genomes can come, at least, you know, per type, hymenoptera database, or you choose, or the I5K space, because I'm not saying it's going to be easy. No, yeah, it has some complications, but resources is better if you allocate all in the same block.
Got it. So, and briefly, maybe can you please tell us what are the requirements to publish data on VectorBase? I say that because I think that you work with really heterogeneity kind of data. Mm-hmm. And in terms of users, what do they need to publish data on VectorBase? Maybe five main points to have into account for the users.
So you are saying how to use the data that is in VectorBase and use it for your publication, like how to?
No, to upload my own data.
Oh, okay. So you're right. VectorBase is the home, but we do not produce the data. So if you want your data to be in VectorBase, that's what you're saying. If you want your data to be in VectorBase, all we need is for you to contact us and tell us, you know, my data is already in an archival repository, is in GenBank or is an EMBL-EBI. This is my accession number, and I also wanted my data in VectorBase. So again, we are not archival of the three main ones. And from there, we pick up the data. So you don't have to format again. You just provide us the accession, and from there we take it. That being said, I know there is not databases for everything. For example, if you are going back to the question of Carlos Alberto, that he was saying, I want variation data for insecticide resistance in Aedes aegypti. I'm not sure where else you can deposit the data. So if there is not a place where you can deposit the BCF file, definitely VectorBase will take it, even though you don't have it in another place.
Okay, thank you. So we have another question from Luis. Are there some problems using unpublished data from VectorBase or from those databases that you show us?
This is what I said at the end of the presentation. The reason why we have the database is for you to use it. So we want people to, if we are just right there for you to look at it, but you do not publish with it, we are not doing anything. So yes, please use the data, but remember what I said. So here, for example, are the genomes. I hear echo, Jesse. Okay. So this is the list.
Now it's okay?
Yeah. Now it's okay. Thank you.
Okay.
For example, these are all our 40 genomes. So you need to check for the genomes available in VectorBase, which one of these is being published or not, and follow the recommendation of the Fort Lauderdale Agreement 2003. And if the data has been published, you can do whatever you want. If the data has not been published, you need to contact the consortium group that is sequencing and ask for permission to work with the data. But please, yes, use it as much as you want, screenshots and images and anything.
Okay, thank you, Gloria. So people are asking me about the presentation, if it's possible to share, but I don't know. It's up to you.
It's going to be in the YouTube video anyway. So.
Yeah, I say the same thing. So guys, we are going to upload this video in the next 2 hours, and then you can check again the presentation and to watch again the slides. So maybe for this time it's not possible to share, and in every webinar that we are organizing, we are not sharing presentations. Okay, please go to YouTube channel and enjoy. So Gloria, again, thank you, and great talk, and thank you very much.
No, you're very welcome. Thank you very much for the invitation. And if you have any questions about anything, please feel free to email me. We'll be more than happy to answer all the questions.
Great.
Bye.
Bye.